Okay, imagine this. A piece of fabric, maybe something you'd use as a bandage or even wear. Pretty ordinary, right?
Mm-hmm.
Now, what if that wasn't just fabric, but a sophisticated structure designed to heal you from the inside out?
Right.
What if it could literally become a building block for regenerating damaged tissue in your body?
That pivot from the everyday to the extraordinary is exactly what we're diving into today. We're exploring the incredible convergence of, well, traditional textile innovation and the bleeding edge of tissue engineering.
It's this fascinating space where threads meet regeneration, creating entirely new possibilities for healthcare.
Absolutely.
And to make sense of this, we've been deep in the source material, a truly insightful topical review in biomedical materials, especially that Ghosh 2025 paper, plus a stack of related research.
Mm-hmm. A lot to unpack there.
Definitely. Our mission today is to pull out the most important insights and maybe, a few surprising facts about how textiles are becoming these revolutionary tools for healing.
From simple wound care right up to potential organ regeneration.
Exactly. So let's start by looking at how fabric even got involved in medicine in the first place.
Okay.
It turns out textiles have been with us in healthcare for a long time, but in very humble roles initially.
Right. The sources trace it back to simple things.
Really basic stuff.
Absolutely. Think bandages, surgical wipes, basic garments, essential functions, no doubt, but nothing that really hinted at the complexity we see today.
Right. It was fabric used on the body, not designed to integrate with the body.
Precisely.
So how do we get from, a basic bandage to something that can help grow bone?
Well, that's the fascinating evolution the sources detail. It was a gradual increase in sophistication.
Okay.
Textiles started taking on more active roles like antimicrobial fabrics. Then they moved into things like components in implants, orthopedic pins, cardiovascular patches.
Oh, okay.
This growing integration really paved the way for them to become actual scaffolds in tissue engineering.
And now the sources talk about these textiles getting smart.
This is a major leap. We're talking about fabrics integrated with electronic sensors and, well, other technologies.
Right.
The insight here is that textiles provide this flexible, conformable platform to place these sensors directly where they're needed on or even potentially in the body.
What kind of smartness are we talking about?
Well, examples from the sources include fabrics embedded with electrochemical sensors to monitor things like glucose levels or pH, which can indicate infection, temperature sensors for inflammation, even strain sensors to track how tissues are healing under mechanical load.
Wow.
There's even mention of herbal-treated bandages that use sensors to monitor wound healing progress while releasing compounds. It completely changes the game from a static material to a dynamic, interactive one.
That's incredible. The fabric itself is providing data and potentially treatment.
Yeah.
But stepping back, why textiles?
Yeah.
What makes them uniquely suited for this jump from passive material to regenerative scaffold?
The core insight is their inherent structure and versatility. Textiles, by their nature, are built from interconnected fibers. This fibrous architecture is incredibly similar to the body's own extracellular matrix, the natural 3D network that supports our cells.
So it's basically about mimicking nature's design.
Exactly. Textiles allow us to easily create these complex, porous, fibrous 3D structures. You can tune the fiber diameter, the porosity, the alignment.
Right.
This adaptability, using both traditional and modern methods, makes them ideal frameworks to guide cell behavior, something that's, difficult to achieve with just solid blocks of material.
Okay, so the structure of textiles is a natural fit for creating these biological scaffolds. Now let's get into what these scaffolds are actually made of. The sources break it down into different material types.
Right. So for creating these 3D support structures, or scaffolds, you need materials that the body will tolerate and ideally interact positively with.
And the first big category is natural materials.
Yes. And the insight here is their inherent biocompatibility, because they come from biological sources, things like collagen, chaisan, silk-based materials. They're often recognized by the body and cause minimal immune response. They promote cell attachment and growth quite naturally.
Makes sense.
The sources highlight their cytocompatibility and low immunogenicity as key advantages. Silk, in particular, is being reevaluated. Historically used in sutures, its unique properties make it incredibly versatile for complex biomedical textiles.
So the body feels right at home with these, but then we have the synthetic materials.
Synthetics, like the polyester family you see in fabrics, though, different compositions, of course, things like PLA, PGA, PLGA, and PCL.
Right.
They offer a different kind of advantage. Tunability. The insight is you can engineer their mechanical properties much more precisely. They can be made very strong, very durable, making them suitable for load-bearing applications where natural materials might not hold up, like supporting bone growth.
But the sources also mention downsides to synthetics.
Yes. While strong and tunable, they sometimes lack the natural cell binding sites found in natural material.
Okay, weaker interaction.
Leading to weaker cell interactions, yeah. Also, their degradation products can occasionally be inflammatory, and achieving sufficient strength for high-stress areas can still be a challenge.
So you've got body-friendly, but maybe less strong naturals, and strong tunable synthetics that cells might not love as much. That sounds like a perfect setup for collaboration.
It is. The sources really emphasize that the most promising scaffolds are often composites or hybrids.
Okay.
The cleverness here is combining the strength using a synthetic textile structure for mechanical support, then coating it with a natural material to create a cell-friendly surface.
Right, to make it more welcoming.
Exactly. Or blending them to get a balance of properties. This is how researchers are working towards the best of both worlds.
And the materials are getting even more advanced.
Absolutely. The sources mention integrating cutting-edge materials. For example, Emzine, a type of 2D nanomaterial, when added to nanofibers, shows enhanced interaction with specific cell types like bone marrow stem cells. And for bone repair needing serious strength, they're even exploring biodegradable metals, like magnesium alloys.
Biodegradable metals.
Yeah. The insight is that these metals provide robust initial support and then gradually dissolve, and the magnesium ions released can actually stimulate bone cells.
Wow.
It's a scaffold that disappears while helping the bone regrow.
Metal dissolving inside you to help you heal. That's certainly a surprising fact. Okay, so we have these incredible materials, both natural and engineered. How do you actually turn them into the complex 3D textile structures needed for a scaffold?
This is where the genius of adapting traditional textile techniques comes in. You're taking ancient crafts and applying them with modern precision.
Like weaving, for example.
Why? Weaving is all about interlacing threads. The insight is that this allows incredibly precise control over the structure, the tightness, the pore size, the overall shape.
Okay.
And they've pushed it further with 3D weaving to create complex, interconnected pore networks essential for cells to move through. Or microweaving and nanofiber coatings to better mimic the body's own tiny structures.
And knitting, the one that gives you loops and stretch.
Exactly. Knitting's inherent elasticity and flexibility make it ideal for tissues that need to stretch, like blood vessels or skin.
Right.
The insight is that this looping structure provides flexibility not easily achieved with other methods. Again, they're doing 3D knitting for volume and critically functionalizing these knitted structures.
Making them do more.
Yeah, adding bioactive molecules or even integrating conductive materials to create smart knitted scaffolds that can stimulate cell growth for things like muscle or nerve repair.
Braiding sounds like it's about strength.
It is. Braiding creates strong, lightweight structures by intertwining threads. The insight is that braiding naturally creates aligned fibers.
Oh, interesting.
Which is exactly what you need for tissues that experience directional forces, like tendons and ligaments.
Makes sense.
They're using 3D braiding for controlled architecture and multi-layer braiding to build complex composites. And they can functionalize braided grafts too, embedding growth factors like VEGF to encourage blood vessel growth in a vascular graft, or even incorporating responsive materials that change shape or release drugs based on the local environment.
So, taking ancient skills to make incredibly sophisticated medical devices. But alongside those traditional methods, the sources also highlight 3D bioprinting.
3D bioprinting is a more modern additive manufacturing approach. The insight here is the unparalleled precision it offers in building structures layer by layer.
Layer by layer.
You can control the placement of materials and even cells down to a microscopic level.
And that uses bioinks, right? What exactly are those?
Think of bioinks as the printable ingredients for tissue. They are typically hydrogels or polymer solutions, often loaded with cells.
Okay.
The key requirements the sources emphasize are biocompatibility, the body has to accept them, and printability.
Right, they have to work in a printer.
Exactly. They have to flow nicely through the printer nozzle and hold their shape afterwards. Alginate and gelatin are common examples. There's also work with nanocomposite bioinks, adding tiny particles to enhance properties.
And can bioprinting create those fibrous, textile-like structures we were just talking about?
Yes, that's a key point. Techniques like extrusion-based printing can deposit the bioink as fine fibers.
Oh.
By controlling the printer path, you can mimic the aligned fiber architecture you get from braiding or knitting.
Got it.
This is vital for engineered tissues like nerves or ligaments where fiber orientation dictates function. And multi-material printing allows for creating scaffolds with different zones for different cell types or functions.
Like different layers.
Yeah, like building complex vascularized skin grafts or structures for nerve repair.
It's clear that whether it's ancient techniques or cutting-edge printing, crafting the right structure is paramount. And once you have that structure, the next step seems to be making it the perfect welcoming committee for cells.
Exactly. The design of the scaffold isn't just about making a shape. It's about creating an environment that encourages cells to move in, attach, and behave as they would in the body.
Right.
The overarching goal is to mimic the body's own extracellular matrix, the ECM.
And a big part of that is porosity, the holes in the scaffold.
Crucial. The insight is that these holes aren't random. Their size and interconnectedness are carefully controlled or tunable.
Okay.
You need different pore sizes for different tissues. For bone, where blood vessels and large cells need to infiltrate, larger pores, maybe 200, 400 micrometers, are necessary. But for something like a wound dressing aimed at preventing infection, smaller pores, even down to the nanometer range, might be used to block bacteria while still allowing fibroblast cells to migrate.
So pore size is habitat-specific. What about fiber alignment? You mentioned that with braiding earlier.
Fiber alignment provides directional cues for cells.
Okay.
The insight here is that cells follow the lead of the fibers.
Like little train tracks.
Kind of, yeah. For anisotropic tissues like tendons, which are strong in one direction, you need aligned fibers to guide the cells to deposit new tissue along that line.
Okay.
For isotropic tissues like cartilage, where forces are more uniform, a random fiber structure is better. Textile techniques like warp knitting and braiding are specifically designed to achieve this controlled alignment.
And the structure needs to be strong enough to function where it's implanted, right?
Absolutely critical, especially for load-bearing applications. The scaffold needs mechanical properties, stiffness, strength, elasticity that match the native tissue.
Okay.
The sources discuss the problem of stress shielding, where a scaffold is too stiff and prevents the surrounding bone from bearing load, which weakens it.
Oh, right. I've heard of that.
Getting the right mechanical match is vital. They give examples like bone kneading and elastic modulus in the range of gigapascals GPa,
Huge numbers.
while softer cartilage is in the megapascals MPa. Techniques like core sheath electrospinning or adding reinforcements like carbon nanotubes help achieve these properties.
And beyond the physical structure, you can add chemical signals too. You mentioned that briefly.
Yes, that's another layer of refinement, chemical functionalization. Coating the scaffold or embedding molecules is like adding biochemical instructions.
Little messages for the cells.
Exactly. The sources mention adding bone morphogenetic proteins, BMPs, to tell stem cells to become bone cells, or vascular endothelial growth factor, VEGF, to recruit cells that form blood vessels. It's tailoring the scaffold's surface chemistry to influence cell behavior.
So you have a perfectly crafted, chemically tuned structure. Now the real challenge, getting the cells to actually move in and, integrate.
Cell seeding is a significant hurdle. Just dropping cells onto a porous scaffold called static seeding often only gets them on the surface.
Right, not deep inside.
Exactly. Getting them deep inside and distributed evenly, especially in larger scaffolds, is difficult.
How are researchers solving that?
The sources highlight dynamic seeding techniques. The insight is using movement like perfusing cell suspensions through the scaffold using bioreactors.
Oh, like pushing them through.
Precisely. It helps push cells deep into the pore network, leading to much more uniform distribution throughout the 3D structure compared to static methods.
And making the surface itself attractive. Does that help?
Surface modification is crucial. The cells need something to grab onto. Techniques like plasma treatment or embedding nanoparticles can alter the scaffold's surface chemistry to enhance cell adhesion and spreading.
Adding those biochemical welcome mats you mentioned earlier must help here too.
Absolutely. Coatings of extracellular matrix proteins like collagen or fibronectin provide signals that cells recognize.
Familiar ground for them.
Exactly. The sources point to using specific short protein sequences like RGD peptides. One surprising finding was a study showing RGD peptide coating on a knitted scaffold led to a 3-fold increase in osteoblast bone cell attachment compared to an uncoated scaffold.
A 3-fold increase. That's huge.
It's a huge difference from adding just a tiny molecule.
And the scaffold design itself helps with seeding.
Yes, it comes back to design. Porosity allows cells to penetrate, and fiber alignment guides their movement and orientation as they attach and spread, particularly important for building aligned tissues.
What about mimicking the complexity of real tissue with different cell types? Is that possible?
That's where co-culture comes in. The insight is that real tissues aren't made of just one cell type.
True.
By seeding multiple cell types together on the scaffold, for example, bone-forming cells, osteoblasts, and blood vessel cells, endothelial cells, for bone engineering.
Okay.
You can create a more complex environment that promotes integration and function, like getting blood vessels to grow within the developing bone.
And growth factors boost that teamwork.
Precisely. Adding specific growth factors into these co-culture systems acts like a supervisor, accelerating the proliferation and guiding the differentiation of the different cell types towards building the target tissue more effectively.
And even physical forces can influence cell behavior on the scaffold. That seems counterintuitive.
Yes, dynamic mechanical stimulation, often applied within bioreactors, is another key factor. The insight is that cells respond to physical forces.
They feel their environment.
Exactly. By mimicking the mechanical environment they'd experience in the body, like the pulsatile flow blood vessel cells feel or the compression bone cells feel, it encourages them to attach better, align, and produce the extracellular matrix needed for the new tissue.
It's incredible how many pieces have to come together. The right material, the right structure, the right signals, the right mechanical environment. So where are these tissue-engineered textiles actually being applied in the real world? Or getting close?
Well, one major area is advanced wound care. These are far beyond simple bandages. They're textile scaffolds designed to actively promote healing.
How do they do that exactly?
They provide structural support for cells. They can manage wound fluid sources, mention gelum hydrogels absorbing up to 10 times their weight.
Wow, that's a lot.
And they can integrate active compounds like antimicrobials, silver, herbal extracts, even nanoparticles like zinc oxide or titanium dioxide to prevent infection.
And accelerate healing itself.
Yes. Materials like chitosan and alginate are used for their hemostatic properties, helping stop bleeding. For chronic wounds, like stubborn diabetic foot ulcers.
Which are a huge problem.
A huge problem. Scaffolds incorporating MMP inhibitors, enzymes that can hinder healing if overactive, have shown significant results, like a twofold improvement in healing rates compared to standard care.
That's life-changing for many patients. That ability to actively intervene is powerful.
Oh.
What about replacing skin?
There's exciting work on artificial skin grafts. The goal is regeneration, not just covering a wound and minimizing scarring.
Dope.
The sources mention tri-layered scaffolds that mimic different layers of skin and using bioactive modifications incorporating things like hydrosin, hyaluronic acid, HA, and epidermal growth factor, EGF. One specific finding highlighted was an EGF-HA scaffold achieving 50% faster wound closure and notably reduced scar tissue formation.
Faster closure and less scarring. That's key.
Definitely. Woven or knitted scaffolds using materials like PLGA combined with elastin are designed to mechanically mimic the elasticity of the dermis. And incorporating skin cells, keratinocytes, and fibroblasts into these textile scaffolds significantly enhances the regeneration process.
Moving inside the body now, textile structures are also proving vital for vascular replacements, right? Blood vessels.
Yes, creating functional small-diameter blood vessels is a major challenge. Textile-based vascular grafts, often woven or knitted from materials like PT or EPTFE, offer alternatives to traditional approaches which have limitations.
Right.
The insight is that you can functionalize the textile surface to reduce common issues like blood clots.
Like adding heparin, perhaps?
Exactly. Surface modifications with things like PEG or heparin have shown improved patency rates, meaning the vessel stays open over 85% in some preclinical studies.
That's pretty good.
Even more advanced are treatments like NO-releasing textiles, which have demonstrated a 60% reduction in platelet adhesion and a 50% increase in endothelial cell coverage, helping the body accept the graft.
So preventing clots and encouraging the body's own lining.
Precisely. Elastic proteins, ELPs, are added for flexibility, and hybrid grafts combining synthetics with natural materials like collagen improve bioactivity and reduce intimal hyperplasia, a common cause of graft failure. Addressing issues like calcification using compounds like bisphosphonates is also an active area.
And the ultimate vision, engineering complex organs. Where are textiles fitting in there?
Well, the sources show how textile scaffolds are being adapted for that ambitious goal. For liver tissue, silt fibroin scaffolds with specific microscale fiber alignment are used to guide liver cell function, helping maintain over 80% of normal liver-specific enzymatic activity in studies. For kidney tissue, braided or knitted scaffolds are being explored to build vascularized renal units, attempting to mimic the kidney's complex filtration and reabsorption functions.
Wow.
With reported reabsorption efficiencies up to 70% of native tissue levels.
Building something as complex as a kidney unit using fabric structures
Right.
that requires getting blood vessels in there, which seems incredibly difficult.
Vascularization is indeed one of the biggest challenges in engineering larger, thicker tissues and organs.
Yeah.
The sources highlight efforts using angiogenic scaffolds loaded with growth factors like VEGF and PDGF, leading to a threefold increase in capillary density in studies.
Okay, encouraging vessel growth.
Right. Combining textile scaffolds with microfluidic designs and perfusion bioreactors is also key to creating the necessary intricate blood vessel networks to keep the engineered tissue alive and integrated. Reducing inflammatory responses using surface modifications and hybrid materials is also crucial for successful long-term implantation.
It's clear this field is innovating at a rapid pace. Looking towards the future, what's on the horizon for tissue-engineered textiles? What's next?
Well, building on the smart textiles we discussed, the next step is textiles that aren't just passive sensors, but actively sense and treat.
The fabric becomes the pharmacy.
Essentially, yeah. Imagine a wound dressing that detects a change in pH or temperature indicating infection, and then automatically releases an antibiotic embedded in the fibers. Or a scaffold that detects stress and releases factors to promote tissue repair.
That's remarkable. What else are researchers focusing on?
Biodegradability is a major focus, designing textiles that safely and predictably degrade as the new tissue replaces them.
So no second surgery needed.
Exactly. The insight is this removes the need for a second surgery to remove the scaffold. It's also driving more environmentally friendly practices, with sources mentioning exploring sustainable materials, like those derived from food waste, like carrot or orange peels.
Really? From food waste?
Yeah, and using water-efficient manufacturing.
And nanotechnology keeps getting integrated, I imagine.
Yes, non-toxic nanomaterials continue to be crucial. The insight is they can enhance scaffold properties, improve cell interaction by better mimicking the nanoscale roughness of the natural ECM.
Right, that mimicking theme again.
And even guide stem cell differentiation just by their physical presence or incorporated cues.
And how is AI playing a role in all this?
Artificial intelligence and machine learning are accelerating discovery significantly. AI can analyze massive data sets on materials, structures, and cell responses to find optimal scaffold designs far faster than traditional methods.
Wow.
The exciting possibility the sources mention is AI even suggesting customized scaffold designs based on an individual patient's specific data, their genetics, their healing profile, potentially leading to much higher success rates for these complex therapies.
So potentially a personalized, AI-designed fabric scaffold for your specific injury or need.
That's the potential.
Yeah.
Other intriguing possibilities mentioned include directly embedding stem cells or growth factors within the fibers during fabrication, developing self-repairing materials, and further pushing customization through advanced 3D bioprinting techniques.
Okay, so wrapping this up, we've taken a really deep dive into how textiles have transformed from basic medical supplies into these incredibly sophisticated, active, regenerative biomaterials.
It's a story of bringing together diverse fields, material science, engineering, biology, powered by innovations in how we select materials, craft structures, design for cell interaction, and deliver therapies. It's quite remarkable.
And what's so compelling is that this isn't just abstract science. This is about breakthroughs that are creating real-world solutions for major health challenges, improving wound healing, offering better implants, and potentially leading to the regeneration of complex tissues and organs. It's knowledge that directly impacts health and recovery for, well, potentially all of us.
It's truly a field where the ingenuity of textile design is being harnessed for biological repair. A really fascinating intersection.
Absolutely. Considering how textile design is influencing the creation of tissues that feel and function like the real thing, and that we're now looking to AI to design these structures, I wonder how else might we look to the fundamental design principles found throughout nature, from how a spider weaves its web to the structural efficiency of a leaf to inspire the next generation of medical solutions.